Good evening. I would like to thank the Sky Foundation and the event organizers especially for this women's event and for inviting me to share my work with you today. Just to give you a little bit of background on who I am, I'm originally from the East Coast. I went to Cooper Union for a degree in chemical engineering, then the medical scientist training program at SUNY Stony Brook, where I worked under Dr. Howard Crawford, who was there at the time. There, I gleaned expertise in mouse modeling of pancreatic cancer and the important role of certain signaling pathways in this disease. I then decided that I really liked programs that were many years in length, so I went to Michigan for my residency and fellowship training in GI in their research track pathway, which has been one of the best decisions of my life. There, I was reunited with my old mentor, Howard Crawford, and worked in the lab of his close colleague, Marina Pasca-Damagliano, whose lab focuses on the tumor microenvironment in pancreatic cancer. During the past 2 years, I was able to establish a new pipeline to study human pancreatic tumors using endoscopic fine needle biopsies, and I'll be sharing these exciting findings with you today. And of course, another plus in moving to Michigan was that it put me in touch with the Sky Foundation, which awarded a grant to fund some of the work that you'll see today. The first thing that I set out to do at Michigan was establish a rich pipeline for tumor tissue collection. When a patient gets referred to us to obtain a tissue diagnosis for a new pancreatic mass, we consent them for our study, and in collaboration with our interventional endoscopists via endoscopic ultrasound, a minimally invasive procedure where we enter the stomach, the duodenum, and the first part of the small bowel, we identify the tumor and use a needle to obtain 2 passes of tumor tissue for research after the clinical specimens have been obtained. What we get are these small pieces of fresh tissue to do our research. After we digest this tissue, we analyze the cells using a variety of cutting-edge techniques to capture the cellular heterogeneity of each tumor. Specifically, we perform single-cell RNA sequencing, which gives us the entire transcriptome of each cell, and mass cytometry, which gives us information on the surface proteins on each cell. We are also able to make organoids from this tissue, which are 3-dimensional cell cultures made from tumor cells that can be used for drug testing purposes. This work was recently published this past year in Nature Cancer. Here we show that single-cell sequencing from fine needle biopsies results in high-quality data, where each dot is a cell and each cluster of dots represents a cell population. In each of these cells, we can obtain the entire transcriptome of each cell and really delve into these cells on a molecular level. These UMAP visualizations show that we have captured over 30,000 cells from 11 endoscopic fine needle biopsies of human pancreatic tumors. In addition to these single-cell studies, we can also take these small bits of tumor tissue and grow organoids, which are 3-dimensional cultures made from tumor cells that can be used for a variety of purposes in research. One important application is that these organoids serve as patient avatars and can be used as a platform to test which drugs would be best matched to which patient. Going forward in my work, I'll be embarking on a project to study pancreatic cancer patients over time and studying their matched biospecimens before and after chemotherapy. Using the techniques that I showed you, I will map the biology of these tumors to figure out what chemotherapy is actually doing, and using this data in parallel with organoids to see if we can find patient-specific drug targets. I'll follow these patients over time clinically with the goal of correlating tumor biology to clinical course and ultimately aid in personalized treatment decision-making, which, as all of you know, is an unmet urgent need in pancreatic cancer. I'll switch gears for a moment and tell you about another project I'm working on with Dr. Richard Kwon on pancreatic cysts. Pancreatic cysts, which are discovered with increasing frequency incidentally due to widespread use of imaging, represent the lone detectable early precursor to pancreatic cancer, and therefore there is an important role for early detection here. Unfortunately, our current ability to correctly identify malignant cysts is limited. The accuracy of current CT scans in detecting malignancy is only 48 to 60%. Together with Richard Kwon, we are developing a battery of tests using AI analysis of CT scans to develop 3D rendering of pancreatic cysts. As a complement to this, we are making organoid cultures from the cyst fluid and analyzing the cyst fluid with single-cell methods in order to develop a precision tool to identify which cysts harbor malignant potential and need to be surgically resected, and which cysts are benign and do not need resection. Coming back to this slide, I'd like to end my talk at this women's event by telling you about a very special woman in my life, my mom. A CT scan revealed that she had metastatic pancreatic cancer. I'm honored and privileged to partner with people like Sheila to work on pancreatic cancer in any way I can as part of her legacy and the legacy of those taken by this horrible disease. Thank you and enjoy the rest of your evening.